Okay, this is a video overview of the third milestone in the Unit 2 project, that is the effects of climate change on plant phenology. And if you recall, the entire point of this project, all three milestones together, is that you were assigned, each student was assigned a separate flowering plant species. And your mission is to figure out whether, number one, the climate is changing in the place where these plants are growing, or your plant is growing, and then secondly, if the climate is changing, how is that affecting the life cycle of these species? The time that they start flowering, the time that they stop flowering, things like that. So in the unit, or the milestone 2 of this project, we looked at whether the climate was changing at this one location where these 4 species grow, and that's the Rocky Mountain Biological Laboratory in Gothic, Colorado, shown here. And remember, over the winter, the land is covered in snow, and it snows for a very long time, and it is covered in snow for a very long time, all the way from November through April. That snow melts into the spring, the plants start growing, and then eventually flowering. They die back in the fall, that's August through October, I should say. They flower over the summer, that's June and July, and then the snow comes back in late October, November, something like that. So we were, last time in class, we were investigating whether the climate has changed in this area, things like snowfall, temperature during May, temperature during winter, the snowpack, things like that. And so what we discovered was that both snow and temperature have been changing at the research site over 44 years. Less snowfall, less snowpack, the snow was melting earlier, the temperatures were higher both over the winter and in May, so pretty consistent effects across 44 years. Okay. And then what we did at the end of last time was we created hypotheses about how the plants at this site might be reacting or responding to those changes. Would they flower earlier? Would they flower later? More flowers, less flowers, things like that. So in the milestone 3 of this project, you are now going to test those hypotheses to see whether you were correct, whether their flowering changed in the way you thought it did, or if it changed at all. So that's the milestone 3 of this project that you will be doing, the effects of climate change on plant phenology. Remember, phenology means the timing of an organism's life cycle. Like I've been saying, the timing of when a plant starts reproducing or stops reproducing, that would be phenology. The time when it starts growing out of the ground or dies back, that's also part of its phenology. So you're basically going to do 3 things for this last milestone. First of all, there's a data set that is specific to your plant species, and I'll introduce that data set in a second. You're going to use that data set to make some graphs and answer some questions, and in doing so, complete a worksheet that tells you which graphs and questions to answer. You're going to take your answers to those questions and write a lab report style paper discussing what you found. So that's going to be the final project. That's what you're actually turning in for this milestone, is a scientific paper that looks much like the lab report that you already wrote for BSc 2011 lab earlier in the semester. So let's introduce, first of all, the data that you're going to be analyzing and where that data came from. So this data came from a group of researchers led by a biologist, David Inouye, who lives in Colorado. And he collaborates and works with a team of researchers, among which are a couple of researchers at FSU that I also work with. And he has been taking data on the flowering of these plant species in Colorado now for the last 44 years. So we've analyzed the climate data for 44 years, and we have matching flower data to go along with that. And what these researchers have been doing is going back to the same plots of land over that amount of time and recording what is flowering, how many flowers it's producing, and then when those plants stop flowering. So we have this 44-year data set where every summer the same type of data is taken, what is flowering, how many flowers are being produced. So to orient you to that a little bit more here, this is an aerial view of the lab. So in this picture, let me go back here for a second. This picture, this mountain right here would be down here on this image. Right here's the lab itself. You can see the road, and here these would be the cabins you saw in that last picture. So basically, in these fields right here, so some fields in this area up the hill from here, which will be off of this picture, these areas around here, that's where these plots would have been set up. And so these plots are 2 meter by 2 meter, so not very large, but there are 30 of them scattered throughout this landscape here. And the researchers, like I said, would go out at least twice a week to every plot and simply measure how many plants are producing flowers and how many total flowers are there on that plot. Okay. And using that then, for any one plot, we could make a curve of the number of open flowers in that plot as the season progresses. And this would be the phenology, right? When the plants are starting to flower, when they're stopping flowering, when they produce the most flowers, things like that. So here's an example of a curve that we could make from one of these plots. And you will be making one of these curves, but I'm showing you this because these are the variables you'll be analyzing potentially in this milestone. So let's make a curve, say, for this species right here. This would be closer to something that delphinium would be doing, for those of you that are studying delphinium. And that will start flowering in, let's say, in this plot right here, early June, around June 1st. Right, it produces more and more flowers, kind of peaks around mid-June, let's say June 15th, and then starts producing fewer and fewer flowers until it stops flowering in late June. On this graph, that's June 22nd. So using a plot like this, we can define several variables here. Right, we can say, okay, when was this plant first flowering in this plot? That'd be the first flower day of the year. We can say when did it stop flowering in that plot? That would be the last flower day of the year. We can say what were the most flowers we ever saw at one time in that plot, and that would be the peak number of flowers. And we can also say when was that peak, right? In this case, it would be around June 15th, so it would be the date of peak flowering. And then finally, we can measure the flower duration. That was the total length of time the plants in this plot were flowering. So basically be the last day minus the first day. That'd be the total number of days this plant was flowering. So these are the variables that you will be looking at, and you're going to see basically, are these things changing over time? We hypothesize that maybe the plant would start flowering earlier. So the first flower day of the year might be earlier as we go from the 1970s to the 2010s or 2020. Okay. Maybe the last flower of the day of the year is also moving. Maybe there's more or less flowers at the peak. So these are the things that you're going to be analyzing to address our hypotheses. So let me then introduce you to what this looks like in terms of numbers on a data sheet. So available on the Canvas course site, and there's also a Google Sheets version of this, is this spreadsheet right here. And first of all, you'll see that there are 4 tabs on this spreadsheet, one for each plant species. So make sure you're on the correct tab. You should know what plant species you've had, because that was what you did for the first milestone, whether you had delphinium or oridron, helianthella, or lupinus. You're going to choose which of these data sets you're going to use. Okay. Now on the data set, there are several columns shown, and there's a key over here that says what exactly those variables are, but they're basically what I just went through. So here's the year that a specific, let me go to this column first. These are the plots that I was describing earlier, so number 5, number 1, number 3, etc. And here's the species that was analyzed. And then here's the year that plot was measured. So you can see the year in this case goes all the way from 2017 down to 1974. So that's 44 years worth of data there. Okay. For each plot in each year, it says the first flower day of the year. That's this column. Again, the day of the year when the first flower is opened. Here's the last flower day of the day of the year when the last flowers were visible before they stopped flowering. here's the peak flower count, the maximum number of flowers observed at any one time during the summer in this plot in this year. Here's the peak flower day of the year. Remember, by day of the year, that means, starting with January 1st, that'd be day 1. December 31st would be day 365. So this is the number in order day of the year. So this would be probably around, May, June is what we're talking about here with these numbers. Okay, so the smaller numbers will be earlier in the year, larger numbers will be later in the year. This is the peak. This is when the day of the year they had the most flowers at once. And then finally, this is the flower duration. Basically, this number minus this number, it's the total number of days the plants were flowering in a plot in a year. Okay, so again, there's a key here to help you with that. Now, what you're going to be doing is, like you did in the last milestone, making some scatter plots and seeing how these things changed over time. So, for example, maybe we want to know, are the plants flowering earlier in the year? Or I should say, do they start blooming earlier in the year? So I can do a scatter plot here, like we've been doing in this class, right? And say, here's the year, and then I hit Control or hold down Control, and I look at the first flower day of the year. I go to Insert. I'm just making a scatter plot here, as we've done before. Make myself a scatter plot, and then I can add a trend line to that scatter plot as we've been doing. So there is a slight negative trend there, which might indicate that this plant is flowering earlier in the year. Now again, you want to make sure that you are using the correct axis over here. We're not using much of this graph. It might be better if we change the axis. So I can click on this axis, for example, and say move over here. I want the minimum maximum, instead of the minimum being 0, let's say the minimum is 100. And the maximum is, we'll leave it at 250. Now you can see there's a more substantial downward trend there, which means these days the plants are blooming earlier than they did in 1976, which would be in line with at least some of our hypotheses that we created last time. So that's one example of a scatter plot you might make, and there are other variables you could do the same thing for. Just like we've done before, making a scatter plot, a trend line that says what the trend is, and then interpreting that as to why that's the case. So that's what the data set looks like here. And again, this is available on Canvas or through Google Sheets if you prefer that way. So the final thing you're going to be doing, and you'll be analyzing this data along with doing the worksheet that I'm about to show right here. So there's a worksheet available on Canvas. Gives an introduction, and this might be helpful for you when you actually write your lab report or your scientific report. Here's some information about where that data came from, and here's the instructions. So you're basically going to go through this worksheet and follow the directions. You're going to do some analyses, and just like I went through with that, making the scatter plots, etc. And using those analyses, you're going to test these specific hypotheses. Did they start flowering earlier or later in the year? You tell me. Did they stop flowering earlier or later in the year? You tell me. Did they produce more or less flowers each year? And for that, you might want to look at the peak flower number or the duration to help you answer that question. Now, as I note down here, the answer may be it didn't change over time. It was equal in 1976 to 2020. The line was flat, and that is a valid thing that could happen. So some variables might change, some other ones might not. So I would encourage you to, you don't have to say earlier or later. It could be no change in the variables. All right, once you do that, you're going to write a lab report that should be very similar to your aquatic ecology lab report in BSc 2011 lab. So we'll have the same kind of content, labeled sections, title, intro, methods, results, and discussion. This worksheet tells you what should go into each of those things, but a lot of this we've already done, including established hypotheses, how you tested those hypotheses and your methods, what your results were. You're making the graphs as part of this, and then discussing what happened. So all of this, I've listed item by item what needs to be included in this report on this worksheet. But again, you're going to approach this very similar to how you did your aquatic ecology lab report, including graphs with numbers and specific captions, and extensive interpretation of those graphs, telling me why. If these phenology variables did change, why do you think they changed? And you might need to tie in the climate data into that we analyzed earlier. So I'll leave it at that. If you have any questions, do please let me know, or need access to the Google Sheets data set. Otherwise, hopefully this was a helpful introduction to milestone 3 of Unit 2 in BSc 2011.